Welcome back. So what we're going to do in this set of videos is we're going to talk about protein function. And generally what textbooks do in protein function is they basically give you a set of proteins that encompass a wide variety of functions, and that they go into the individual protein's functions. So in the textbook that I'm using, that I teach from, this is Leninger's Principles of Biochemistry, 6th edition. They generally do three sets of proteins. The first kind, they're doing oxygen-binding proteins, which is a very common function. Then they go into immunoglobulins, and then finally into muscle proteins. So out of the oxygen-binding proteins, the main ones we're going to go into here in these next few videos are going to be myoglobin and hemoglobin. And this is going to be the first real playlist in this whole set, and you can certainly go look at other videos as well. We have some on glycolysis, but in these videos we're just going to do protein function. And at this point, what I'm assuming is that you already have a base knowledge of amino acids, their structure, their properties, Lewis acid-based chemistry from your organic, and then also basic things like first, second law of thermodynamics. Anything that you've seen from Gen Chem and organic, I'm assuming that you already have. If we need to go over things, say from inorganic, we'll do that as we go. And in fact, we'll see some things from inorganic in these next few videos. Okay. So this first video is going to be on myoglobin. Okay. So this first video is on myoglobin. And myoglobin, you probably haven't heard as much about myoglobin if you're a biology major. You would have heard of hemoglobin extensively in a physiology class. And hemoglobin is usually what we're talking about when you're talking about oxygen delivery. But it turns out that for oxygen binding, myoglobin and hemoglobin both do that. Now, when we're talking about oxygen delivery, first we need to talk about the general functions of these proteins. Hemoglobin does not bind oxygen as tightly as myoglobin does. So therefore, if you don't bind oxygen as tightly, hemoglobin is going to be a lot better for the delivery. Contrastingly, myoglobin binds oxygen significantly more tightly than hemoglobin. So myoglobin will bind the oxygen molecule, and it will not let go near as easily. Okay. Now, one thing that's important to understand about oxygen-binding proteins is generally they're going to have a heme cofactor, and the identity of the heme cofactor changes depending on which oxygen-binding protein you're talking about. Another oxygen-binding protein that's of particular interest to biochemists is called cytochrome P450. This particular protein is an enzyme. We'll talk more about enzymes in the future. Enzymes are proteins that catalyze reactions by lowering the activation energy. P450 is an enzyme. However, myoglobin and hemoglobin are not enzymes. Okay, they're simply oxygen-binding proteins. Okay, now in the context of myoglobin and hemoglobin, one of the coenzymes that they have, in fact, it's the only coenzyme they have, is called heme B. So there are multiple hemes. This is the particular heme that they carry, and if you notice in the center there's an iron atom, and specifically it's in the +2 state. So we would say that iron is in the ferrous state, right? So iron is in the ferrous state, right? And that means it has a +2 charge, and this is going to be the Lewis acid to which the oxygen binds. Okay. Now what we're doing is we're looking at the heme B molecule, and if you notice, the first thing that should really pop into your mind is that the entire ring, this entire porphyrin ring, is aromatic. Okay, so this is a very, very stable ring. It's going to be difficult if you want to catabolize it. There is an enzyme that does that. It's called heme oxygenase, but it's going to be very difficult to catabolize it. Okay, so if you notice, each one of these nitrogens here that's in the center. Okay, I'm trying to draw lone pairs. Okay, each one of those nitrogens in the center is holding that iron in place. Okay, so if you took this heme, it's going to be very difficult to remove those hydrogens. Remove the iron, right? And the reason is because you have a very stabilizing effect with these nitrogens that are chelating the iron. Okay. So what is chelation? Well, for those of you that have taken an inorganic chemistry class, you would have talked extensively about chelation. Let me write that. This is a chelation. And chelation is essentially a stabilizing effect that is observed when you have Lewis bases that are in a coordinate covalent bond with a Lewis acid. Okay, so at this point, again, I'm assuming that you already have a base knowledge of Lewis acid-base chemistry, but suffice it to say for now, for a Lewis base, you can effectively think of them as nucleophiles, but one of the requirements for a Lewis base is that they have a lone pair to donate the electrons, right? Because the definition of a Lewis base is an electron pair donor. And effectively in this molecule, the nitrogen atoms in each of the pyrrole rings, the nitrogens have lone pairs and they're donating them to the iron atom. And so the Lewis acid, by definition, has to be an electron pair acceptor. Okay, that's the definition of a Lewis acid. And there are several ways you can identify Lewis acids. In general, Lewis acids can have multiple characteristics. Number one, normally when you're looking for a Lewis acid, they're going to have a positive charge, whether it's partial or a full positive charge. In the case of something like this, I mean, this isn't a particular biomolecule, but you notice that that particular carbon in the center has a full positive charge. So when you look at it, it has an empty p orbital. So if you have a nucleophile, nucleophile would do nucleophilic attack on that carbon, right? So you can look at things that have full positive charges, and sometimes you can even have things like this. Sometimes if you have something like this, something with a good leaving group, also something that has a partial positive charge because there's a dipole moment right here, that's also prone to undergoing or being hit up by a nucleophile for nucleophilic attack, right? So things that have full positive charges, partial positive charges, those things tend to be good Lewis acids. Now, if we come back here for the purpose of this, iron has a full positive charge, so it definitely is the Lewis acid, and it's accepting electrons from each of the nitrogens from each of the 4 pyrrole rings. And so, for that reason, we call these hemes tetrapyrroles. And each one of the subunits of hemoglobin and each myoglobin molecule have one of the hemes. Now, if we look at the heme molecule itself, okay, what we notice is that it's fully aromatic. Okay, so each one of these lone pairs on the nitrogen, what you can do, and I'll try to do my best to draw it with space, is you can draw a resonance structure, and you can certainly see that you can move these electrons throughout this entire molecule, and I could go on and on and on with this, but suffice to say that you can, the whole ring is aromatic. So as a result, the porphyrin ring, heme B, and all the other hemes are planar, right? They're planar. So what we're doing right now is we're looking at the heme, we're looking at it face on, okay? So typically when we abbreviate heme, what we'll do is we'll say, okay, well I have 4 nitrogens, right? And then I just have the heme in the center. So they're in there chelating this iron atom. And the type of, and we draw them with these single bond looking bonds, but they're actually not single bonds. What they are is they're called coordinate covalent bonds. And if you take an inorganic, that probably makes some sense. And coordinate covalent bonds are basically what are involved in chelation. Okay? Chelation, as we said, is an electrostatic interaction between a Lewis base and a Lewis acid. It's not a reaction, but it is an interaction. Okay? And essentially what you have is these coordinate covalent bonds, and they're not covalent bonds because they're weaker than covalent bonds, but they're stronger than ionic. So they're somewhere in strength between covalent and ionic. But effectively, because the nitrogen atoms aren't moving because of the stability of the porphyrin ring, the iron atom is basically held in there very stably. Okay, so this right here, this setup right here is if we're looking at the porphyrin ring face on. But if you imagine if we have an axis right here, and I was to rotate it 90 degrees. So in other words, do this in a different color. If I have an axis that runs through here, right, and I rotate it 90 degrees, then what you basically have to see is that the nitrogens, I'm going to do my best to draw it, but you have your iron, right? Right? I'll just do it like this. Now you're looking at it flat, right? So if you look at it sort of from the side, essentially these 2 nitrogens right here would sort of be next to each other because the ring's planar, right? But effectively the whole ring's flat, right? So what you have to think about is that in this porphyrin ring, okay, these nitrogens are occupied by the porphyrin ring, but you have to imagine that Also, you could potentially hit something from the top here, so from this side, and from the bottom, right? So something can interact with this iron, okay? And that's in fact the case, okay? So what you have to realize is that you have some part of the myoglobin, because we're talking about myoglobin here, the protein chain, right? So here's your protein chain right here, and you have to imagine that there's residues sticking off of the chain. One of the critical residues that we have is a histidine. So what you're going to have is a setup like this. So you're going to have, okay, did my best to draw it there, but essentially you have to imagine that your imidazole ring of the histidine sticking off right there. And if you remember, on this particular nitrogen, you have a lone pair. And this specifically, I didn't draw it really in the right orientation, but you have to imagine that this imidazole nitrogen is interacting with this iron, right? So, in other words, that nitrogen of the imidazole ring, the one without the proton on it, that's acting as the Lewis base, and it is also adding to the chelate effect that's holding that iron there in the center. So, also what you have to imagine is that when we look back up here, this part here is exposed, meaning it's exposed to the blood. Okay, so I want to be perfectly clear that myoglobin is not enzymatic. It's not an enzyme. It's not catalyzing a reaction. But you sort of have to view this area up here sort of like the active site of the enzyme. Okay, and so what will happen is and let me draw it like this and come over here. So what you have to imagine is you have this iron, these nitrogens of the porphyrin ring, and then you have this histidine nitrogen, right? This is of the histidine, and this is nitrogen occupying what we refer to as the axial position. Okay, so going up and down like this is the axial position. And so what can happen is myoglobin can bind oxygen. So oxygen will come in, right, like this, right? Oxygen will come in like this, and it will interact with the iron. Okay? Oxygen's lone pair can act as a Lewis acid. Now, the question you might be asking is, why does oxygen come in at an angle like this, right? It doesn't come in perpendicular to the porphyrin ring. It comes in at some angle theta, right? And the reason it does this is because if you look at oxygen, so you look at oxygen, so here's oxygen, okay, diatomic oxygen. You have to remember that lone pairs on oxygen are right here, right? It's that way for both of these oxygen atoms, right? So if you remember for Lewis bases, which is what oxygen is doing to the iron, it's the lone pair that's acting as the Lewis base, so that's why oxygen comes in at an angle. Okay? There are other molecules, bad molecules like carbon monoxide. Carbon monoxide has a triple bond, positive charge on the oxygen, lone pair on the carbon, negative charge. So there are other molecules like carbon monoxide that can come in and interact with the iron. But a lot of those come in linearly, right? Because if you picture the carbon with the electron domains, you have one electron domain right here, right? And a second one here. So this setup for the carbon is linear. So if you had carbon monoxide coming in here attacking the iron ion, it would come in perpendicular to the porphyrin ring. But oxygen has these lone pairs that are basically, you could view them as 120 degrees approximately to the double bond. For that reason, it comes in an angle. Okay, so the point now I want to underscore is that myoglobin is binding oxygen. Okay, and it specifically is binding it very, very tightly. Okay, so you can imagine that with myoglobin, myoglobin is not going to want to release oxygen very much. Okay, and in fact, what you'll find is that oxygen under normal myoglobin under normal conditions will not let go of oxygen. Okay, and to understand this, we need to look at Le Chatelier's principle. And one thing I just want to underscore at this point is that really with these proteins, myoglobin and hemoglobin, and with red blood cell function in general, this is really just an exercise in Le Chatelier's principle. So one thing I'll tell you right now is that myoglobin is typically abbreviated MB, right? And so let's say we have free myoglobin plus oxygen, and that's going to be in equilibrium with the complex of myoglobin and oxygen, right? Hope that makes sense. So under normal conditions where we have a normal partial pressure of oxygen in the blood, myoglobin will not dissociate from oxygen. So in normal conditions, it's going to favor this state, right? It's not going to let go of oxygen. But if you imagine if for some reason maybe our hemoglobin gets saturated with carbon monoxide, or maybe we go up into high altitude conditions where there's lower partial pressure of oxygen, right? You have to imagine that the partial pressure of oxygen in the air is going to drop, and therefore it's going to drop in the blood as well. So if you think about it like that, let's think about it this like Le Chatelier's principle. Remember, you can treat pressures like you can concentration. So if the partial pressure of oxygen drops, right, by Le Chatelier's principle, which direction is the reaction going to shift? Right, it's going to shift back this way, right, to relieve the stress, right? If you take out a reactant, at least in the way that I've drawn it here, it's going to shift back towards the reactants, right? Okay, and so there's direct application of this. If you have low partial pressure of oxygen, you give someone an oxygen tank, right? And it raises the partial pressure of oxygen and causes oxygen to be bound both by myoglobin and hemoglobin. So what does this tell you about myoglobin? Well, it tells you that myoglobin, because it binds oxygen so tightly, your means of getting oxygen to cells is not going to be myoglobin. It's going to be hemoglobin, and we'll talk about that later. The only way you're going to cause myoglobin to dissociate oxygen is if you have very bizarre conditions, like if you have carbon monoxide poisoning or you have just high altitude. So normally it's not going to dissociate oxygen, but really it's more acting as an oxygen reservoir. So if you have those bizarre conditions, it's more like a metabolic safety valve, right? If for some bizarre reason your oxygen, your PO2 gets lowered, it's kind of like having a spare tire, right? You don't always use your spare tire, but if you get a flat tire, you have something that you can drive with, right? It's the same way with myoglobin. If you have, for some reason, your partial pressure of oxygen drops, you have spare. And the reason it's called myoglobin is because ordinarily myoglobin hangs out in the muscle tissue. Myo is a prefix that means muscle. So a high concentration of myoglobin you'll find in the muscle tissue. Okay. Now, another thing that's important about myoglobin that we haven't talked about thus far is that it's a monomer. Okay. Myoglobin is what we refer to as a monomer. So when you see myoglobin, it's just going to exist as one single polypeptide chain. Okay. So this is, let me do this. Do it like this. Oops. Let's just put like this, MB for myoglobin. It's just going to exist as a single polypeptide chain, and so it's functional with simple tertiary structure. Okay, recall that tertiary structure is the sum component of the polypeptide chain plus any secondary structures that we have, right, including disulfide bridges and all that good stuff, right? But the point is that myoglobin exists functionally with tertiary structure as a monomer. What we'll see with hemoglobin is that changes because with hemoglobin, you actually have to have quaternary structure. So there's 4 subunits that have to come together with hemoglobin. But for the purpose of myoglobin, it's functional just as a monomer. Okay. Now, we've pretty much hit everything with myoglobin. Now, myoglobin is, like I said, a monomer. Therefore, it does not exhibit a property of hemoglobin called cooperativity. Now, that probably doesn't make any sense to you at this point, but if you watch the video that we'll have on hemoglobin, we're going to explain what cooperativity is, and just suffice it to say for now that myoglobin does not have cooperativity, and the reason it does not is because myoglobin is a monomer. So that's important to understand at this point. Now, what I hope you've gotten out of this video is generally that Le Chatelier's principle becomes incredibly important when we're talking about red blood cell function. We'll have other enzymes that are associated with the red blood cell that operate by Le Chatelier's principle, but the point is that if we have normal partial pressures of oxygen, oxygen will not dissociate from myoglobin. Okay, and then also we have the structure of heme B, which is a coenzyme, or a prosthetic group of myoglobin, and that's what actually binds oxygen. And actually, one thing I will underscore here is that in the case of myoglobin and hemoglobin, there's a histidine residue that chelates the iron from the bottom axial position. That can change depending on which protein you're talking about. In the case of hemoglobin, it too is a histidine imidazole. nitrogen. But in the case of something like cytochrome P450, it becomes a cysteine residue. So the identity of that particular residue can change. So don't just get caught up in thinking that it's always the same. No, it can change. But for the purposes here, we're just going to consider the histidine residue. And just understand that the function of myoglobin is acting as an oxygen reservoir, and it normally will not dissociate oxygen. So I hope this video helped. In the next video, we're going to talk about hemoglobin function, as we'll find hemoglobin is significantly more complex than myoglobin. See you in the next video.